Okay, well, thank you for attending and your patience here. I want to present Aimm Therapeutics. We're a biotech company located in Amsterdam. We select naturally occurring antibodies from cured cancer patients. These are patients typically post-immune therapy, but not always. We have the best-in-class technology for doing exactly this, and I think I'll convince you of that. We have closed a $25 million Series A, a single US investor, back in 2018, and we are racing forward with a lead compound to the clinic. We'll file an IND next year, and we have another one about 18 months behind that we intend to put in the clinic ourselves. Okay, so this is the secret sauce. This is the patented technology in the company. Now, the way I look at it is we are in the information business. The information we want is the DNA sequence of antibodies. If we lose those B cells, we lose that information forever. So what we do is we'll take the memory B cell compartment, essentially the entire immunological history of a patient, and we'll immortalize their memory B cells. We do it with a retroviral transduction of two oncogenes, BCL6 and BCLXL. And this stops those plasma cells, those plasmablast cells, going into apoptosis or differentiating into plasma cells, which we can't culture. Since it's a retroviral transduction, we get about, say, 95% of the entire B cell repertoire. We essentially get all the information from this patient. These cells are so robust, we can freeze them, thaw them. They secrete antibody into the supernatant, allowing us to do functional assays. They have B cell receptor on the surface, allowing us to label antigen and fish them directly out. This is a very robust system. In fact, some of these B cells we have used for about 10 years in and out of the freezer with no lack of any of these properties. So I think the best way to understand our company, given that we select antibodies from cured cancer patients, is to give you an example here. So in 2006, a patient presented and was treated in the University of Leiden with stage 4 melanoma, including brain metastases. Now in 2006, this was before checkpoint inhibitors. This was a difficult diagnosis. The patient was put into an experimental TIL therapy, 1 of 10 patients, the only responder. Complete response. Stage 4 melanoma with brain mets, complete response. We ran into this patient 6 years after their complete cure, got a blood draw, immortalized the memory B cell compartment, all the immunological history of that patient, and began the process of fishing out an antibody that binds and ultimately kills melanoma cells. The question, and this is really what our company was built to do, is ask, did this patient make an antibody that others will benefit from? Does this patient make something that we can manufacture as a product? Here's how we do that. So you see in the top left, we have immortalized the B cells of this patient, this example. You can imagine we put 1 B cell in each well. We allow them to culture. The supernatant gets enriched with antibody, and we do a functional assay. In the case of oncology, we're looking for binding to representative tumor types unrelated to the patient. We're not looking for personalized medicine. And does it not bind to representative healthy cells? That's the initial step we take. Once we have a strong lead, you can imagine we do a great deal of work to resolve this specificity, which is what we're after. When we have an antibody that we like, we characterize it. We do in vitro work and determine that we have something that really is worth investing in. And then we take that antibody and we do immunoprecipitation and pull down the target from the tumor cell. We do mass spec, we knock the target in, knock the target out, and only then do we identify what the target of this antibody is. And once we know the target, we can ask, are there any other tumor types that also express this? So that's a general flow. If you look in the bottom of this cartoon here, the industry really starts to the far left and works backwards. The industry says, I want to be in prostate cancer or lung cancer. I like CDXYZ. Now make me an antibody. And in fact, we do exactly the opposite. We say the immune system liked a target, a tumor-specific target, made an antibody, and it's up to us to resolve, well, what is it that it did and where else can we use that? So I'm going to show you a little data. These in vivo studies are done in mice that have a human immune system. They have B cells, T cells, macrophages, dendritic cells. We make these mice ourselves on site, one of our capabilities. So this is A375. It's a standard melanoma model. Checkpoint inhibitors are approved in this indication. So if you look at this graph, you see nivolumab, a representative checkpoint inhibitor, has significant activity. And yet you'll see that we have very significant single-agent activity from this antibody we pulled out of our surviving patient. And if we add a checkpoint inhibitor on top of that, we get a little extra kick. Now, what's more interesting is if I look at tumor slices here. So the second row is a checkpoint inhibitor alone. The blue arrows, and it may be difficult to see, on the left point to the blue macrophages that cannot enter the tumor. And on the right of that second row, the brown T cells lined up around the outside. They can't enter the tumor. The third row with the red arrows is our antibody alone. There you see massive infiltration of macrophages and massive infiltration of T cells. And when you add a checkpoint inhibitor, when these are angry T cells, you decimate the tumor. And in fact, it was difficult for us to find scraps. Now, this is a standard melanoma cell line, so now I'll show you a highly resistant melanoma cell line, SKML5. Nothing works in this setting. This is the highest bar that we could find. Here you see on the graph, again, a mouse with a human immune system. The top three lines, I would say nothing works. You may be tempted to think that we have some single agent activity there, but I would suspect if we reproduce this experiment, those lines would all move around a little bit. Nothing works. But if you look on the bottom, the combination of this antibody from a cured cancer patient with a checkpoint inhibitor, you flatline this extremely aggressive tumor. If you have worked with SKML5, you'll recognize that is an important result. More interestingly, if I take some of these complete survivors and I allow them to live off treatment, I see that I have what is clearly a CD8 positive vaccinal response. I would assume this is the type of experience that the original patient benefited from. Exposure to the antibody, cell kill, long-term survival. So an interesting antibody, and I think it represents what we're doing. This is our lead. It's in GMP manufacturing. We'll have tox material shortly. We'll do the GLP tox early next year, and we'll file the IND at the end of the year. So what do we see? If you take something away from the presentation, take this point away. We trust that the human immune system has found an antibody that contributes to the clinical outcome of a surviving patient. It's the immune system of the patient that is tolerant to all the proteins on healthy cells. Unless you have autoimmune disease, your immune system is not going to make an antibody response against healthy cells or the proteins on those cells. So what the immune system sees, A, pathogens, it makes many, many antibodies, hundreds of antibodies against complex pathogens. But in the case of cancer, the immune system will see a post-translational modification, something that the tumor cell did that healthy cells don't. It's a slight modification. It could be a truncation, phosphorylation, a sialization. We see in the top of this table, these are examples of proteins that have been modified on the cell surface. We also see modifications of intercellular proteins that are then trafficked to the cell surface, and again, the immune system sees it for the first time. So by leveraging the selectivity of the immune system, we by default find tumor-specific antibodies and novel tumor-specific targets. Now, we are not engineers. We are biologists, immunologists. We do a lot of cell culture. We manipulate the immune system. So if there are tumor-specific antibodies and targets that look like they're investment quality, we are not going to build bispecifics and ADCs and CAR-Ts. There are others with expertise that engineer in function and potency. They do that well. So a program you can imagine that fails in our hands, where we find an antibody that is tumor-specific against a novel tumor target, but we don't think it has the punch to be a product. That failure, in fact, is a solution for the rest of the industry that brings all these heavy weapons to bear, but they're so dangerous, they really need tumor specificity. So that's part of our business model. We'll monetize those. So we will not, to be clear, develop all these compounds. We'll monetize them. What will we work on? Our lead, 1412, is the melanoma program that will be in solid tumors and potentially be ALL, and we'll make that decision at the end of the year. We have another compound about 18 months behind that, 1704, very specific, very potent antibody. That looks like that's one we're going to invest in ourselves. And then we'll keep a couple others kind of in the ballpen, to use an American analogy, a baseball analogy. So if we need to swap something out, we'll have something ready to go, and we will slowly advance those. Everything else we'll monetize. And we're in discussions with companies that are very interested, particularly the CAR T folks, when we show them bispecific CD3 T-cell engaging data. It's very, very convincing. And so we hope to pursue a more strategic deal with one of the big players in that space, but time will tell. I'll give you another example of a different patient. And again, this is what we would call a failure in our eyes. We're not going to take this alone. This will be something that we would out-license. So this patient presented in AML blast crisis at the AMC where we're located. They got a transfusion and a human stem cell transplant. That's a new human immune system. It's an immunotherapy. So that immune system, which had also grown up with CD43, goes after the AML and finds that in AML cells, they make a modification, a post-translational modification of CD43, where they oncosilate it where it should be glycosylated. We've published on this, I think, in Bloods, but well understood. Now, this immune system had to resolve this oncosylated CD43 against this ocean of healthy CD43 broadly expressed in the hemopoietic system. To do that, as you see in the second bullet, required 37 somatic hypermutations. That is a tremendous amount of antigen experience. That is a lot of work in a germinal center trying to mutate that B cell, get an antibody that could resolve both of these. That's the function of the immune system. This antibody that we pulled out binds all AMLs we've tested. Now, we never test against the patient's original tumor. That's personalized medicine. These are over 80 samples from the clinic of patients that have come in AML blast crisis, where we have tested their AML. Every one of them reacts with this antibody. It reacts with AML stem cells, which is how you cure AML. It reacts with all MDS that we've tested, and about 50% of the melanomas. If you see on the bottom left, it's an early experiment, clear, strong, single-agent activity in a humanized mouse. And to the right, you see it very clearly internalizes. So you can imagine the ADC folks who are interested in putting these powerful warheads on antibodies and need that specificity are very interested in this program. What I haven't shown you here is we have a kind of demonstration technology where we make CD3 T-cell engaging bispecifics. We've done that in this program. That data is so convincing that the car companies have asked to build cars. They've built cars. The in vitro data looks good. And we think there's an opportunity here to solve a big problem in that sector of our industry. And we could play a big role there. So we'll pursue that. But this is an example. As interesting as this is, it's an example of something we're not going to work on ourselves. We'll monetize, maybe develop in collaboration. So finally, I've talked about what we're going to be doing ourselves, the non-dilutive cash flow we'll get from partnering, but there's another deal that we've done. This is a deal we did with one of the German Merck's companies. This is Sigma. Sigma is a giant company that sells antibodies to all your researchers. So when you go back to your companies and you can look in your labs, they're ordering from Sigma. Sigma is going to scale up this technology, has scaled it up, and they're going to move their polyclonal antibody business over to monoclonals. We've gotten milestones from them, we've gotten royalties from them. They've hired, I can't tell you how many people, but a small army of people, reproduced our laboratories, and they're selling these antibodies already. That's how robust and scalable the technology is. So that's the Aimm story. I would tell you we're finishing up in 2019. Next year we will be filing an IND or CTA, likely a CTA, and we'll work with the NKI and some of our close collaborators. And we will designate a clinical candidate. Very quickly, it'll likely be 1704, and then that one goes into IND-directed experiments as well. I've asked the company to prepare themselves for an IPO in 18 to 24 months. I think that adds a certain discipline to the company. It makes us available to that exit, and certainly maybe more interesting to someone doing diligence from the corporate side. But we see things at this company that no one can see. We find tumor-specific antibodies like no one else can find, and we think we have a great future. I would finish this by saying we have a new CSO, Hans von Einenam. Hans invented Keytruda, to give you a sense of where he comes from. Hans could have gone anywhere, and he chose to work with this company. And I invite you folks to do the same. Okay, thank you for your time.